Hello, I'm Mike Blatt. I'm the editor-in-chief of Plant Physiology and have been since 2013. And I'm here to talk to you very briefly about our most recent focus issue in plant cell dynamics and about our focus issue program in general. Plant cell dynamics is a fascinating area of research, and it's one that is attracting more and more attention in biology in general, but certainly in all aspects of cellular function, plant function, and how plants interact with their environment. And it's really driven by some amazing advances in technology that have taken place over the past 2 decades. And this now allows plant biologists and biologists in general to access the properties of cells and how they function in real time. And that has opened up huge areas of research that bridge the gaps between plant biology, biochemistry, biophysics, and physical properties of cells, as well as the more traditional aspects of genetics and molecular biology. So bringing all those together has really driven some exciting new advances that no doubt are going to change the way we think about how plants function and how they interact with their environments. So I'm going to pass you over to the four editors who handled the focus issue. That's Dan Zemansky, Diane Bassam, Wataru Sakamoto, and Tun Munich. And you will see they're interviewed by several of our assistant features editors, and they will be able to tell you a little bit about the focus issue on cell dynamics. We run a focus issue program within plant physiology and have been doing so for well over a decade now. Typically, we're publishing between 2 and 4 focus issues each year with the idea of bringing together the most exciting and cutting-edge science and reviews of developments in the field, along with primary research articles that go in that topic area. So these are great resources both for teaching and also for researchers who are interested in getting into an area and learning more about it firsthand through experimentalists who are working in the field and can provide enough information to get researchers off the ground in starting their own studies. And while obviously we're focusing at the moment on cell dynamics, we have a number of other focus issues that are coming up in the near future. For example, there'll be one in this coming year on biotic interactions, and we're aiming for another focus issue on synthetic biology. And you will find information about these already on our website. So if you are interested in submitting articles that would be relevant to either of those, please do have a look at the website or contact the editors to find out more about what they're looking for. So thanks very much, and I'll pass you over now to our fantastic cell dynamicists.
Well, let's start by, Dan, you're a co-editor for a new feature special issue in Plant Physiology focusing on cell dynamics. So tell me a bit about yourself and why you're involved.
I'm a cell biologist at Purdue University. I've been working on cell morphogenesis for 20 years, and there was an opportunity to feature some of the hottest research in the area of cell biology, and so we put together a focus issue on the broad topic of cell dynamics. We're really interested in emphasizing how the cell changed over time, different components in the cellular and molecular mechanisms of shape change and trafficking, cell wall assembly.
Could you tell us something about what is meant by cell dynamics and your particular interest in this topic?
Okay. my interest is, as I mentioned, I'm interested in organelles which derives from endosymbiosis, namely mitochondria and chloroplasts. And those organelles contains more than two membrane systems, unlike the other organelles. And how these organelles develop is very fundamental, particularly for energy producing standpoint. And the chloroplast is very, very dynamic in the sense that it responds to light and then moves mitochondria as well. So this kind of dynamic nature is very important part of cell dynamics.
With respect to the topics that are being kind of covered in this issue, how do you think about relating them to kind of broader topics that might be interesting to non-scientists or communicating it to non-scientists?
I think because cell biology is so fundamental to everything else that happens, and cells are really the defining feature of life. So I think this means that we can relate cell biology and things that are happening in cells to almost any other aspect of biology, including growth and development, and even as far as ecological considerations. We think about stress responses and changes, how the cell biology changes in different environmental conditions.
Mm-hmm.
What are some of the more recent tools that have been developed that allow us to look at cells as dynamic structures?
I think needless to say, most of the articles in this focus issue contain some part of organelle visualization. And I think the most powerful tool which has been invented for the last 2 decades or so is just live imaging analysis. And in addition to that, I think, as I mentioned, the work by Yongyan Jan's work, kind of 3D construction is very important to look at the detailed structure of membranes.
So, I mean, you've had all these advances, but what are some of the challenges that still remain to be addressed in cell biology and cell dynamics?
Well, I think part of it is the image, getting the numbers out. For every hour you spend at the microscope collecting the images, it can be anywhere from 4 to 10 hours of analyzing the images to get the quantitative data out of it. So I think one of the key challenges is image segmentation, extracting out the quantitative information in an automated manner to increase the throughput of the experiments. Because we're talking about time-lapse imaging at the time scales of often hours when plants or cells are slow growing. So the data sets can grow quickly to the gigabyte size, and dealing with that manually isn't the most efficient solution. The other thing is, image registration, and the cells move and tilt, and how do you align the images so that you know where you're at in the cell over time, or that you can properly reconstruct spatial coordinates over time. That's a challenge.
What's your opinion about where kind of the most effort is needed right now?
Mm-hmm. I think quantification is going to be a huge issue. I mean, we have ways to quantify images, but I think there's a lot that still can be done when we're thinking about, fluorescence microscopy, even electron microscopy, and the best ways to quantify and how to know what we're quantifying and what we're really looking at. Mm-hmm. And that's essential, of course, for any kind of predictive work. You have to have accurate and extensive quantification. How is this knowledge going to help us address some of the big challenges that we're facing today? So food security and climate change. So how do we maintain how is this going to help us maintain the productivity that we need?
Let's see. So I think, the next generation, future generations of crops are going to have to deal with unpredictable environments, right, and climate change and new types of pathogens, viruses, bacterial and fungal pathogens. And I think there are major opportunities for engineering plants to have either new architectures or new pathways that make them more adaptable to changing environments or more resistant to different types of pathogens. And these engineering approaches come at a price of understanding the underlying biology. So all of the work that's being done and that's emphasized in this focus issue is trying to understand the key players in plant cells that mediate these important traits. So that's really what it's all about, is building this knowledge base from the ground up so that it can be used to understand traits in a more mechanistic way, to engineer traits rather than just select for them.
Fantastic.